My name's Aquaculture. I'm a faculty member here. We enjoy showing off, and there's a lot of things that have been going on in the past year or two. These tanks over here are some tanks that we set up for experiments on brood fish and diets. The last couple of years, we've been looking at different formulation of diets that would end up yielding higher numbers of fingerling, or excuse me, fry. Last 2 years they've been done in tanks, but right now this year we're planning to do a similar study in ponds to sort of mimic the more applied aspect to this. So these blue tanks up here allowed us to do some experiments with larger fish. The first demo we have is Julianne Jacobs. She's a graduate student of Anita Kelly's, and she's going to be talking about hydrogen sulfide and how to eliminate it, and perhaps eliminate it because it might be a cause of a loss of bait fish.
Aquaculture ponds are ideal conditions for hydrogen sulfide to build up because we add excessive nutrients from the feed and then also from the organic waste from the fish. And so it creates an anaerobic zone once it's decomposed. And this zone usually doesn't come in contact with the fish because they're more in the water instead. But problems seem to occur when during water quality changes, like this time of year when the water starts to turn over because the water temperatures are changing, and also during harvest when they get in with seines and they walk through the soil, and the soil mixes up the hydrogen sulfide into the water. And problems really occur when the dissolved oxygen levels are low, because low dissolved oxygen levels increase the toxicity of hydrogen sulfide, because hydrogen sulfide can be oxidized by the oxygen into non-toxic forms. And so these bottom soils are where they are increased, and then if they come in contact with fish, that's when problems occur. But also when your dissolved oxygen levels are low, typically at night, like into the morning hours, this can cause a problem with fish as well, especially during these times of the year. And so hydrogen sulfide has been linked to massive mortalities. For years there have been large disappearances of fish and large mortalities where fish are just dying. They don't know what's going on. They contributed it to low dissolved oxygen or some kind of water quality problem. And hydrogen sulfide was actually being overlooked, and it was found in the last 20 years that was actually the culprit. And one of the big problems was in catfish industries when it was first discovered was because they used a sinking feed. So as soon as they put it in the water, it floated to the ground, and most of it was not ever eaten. And so that created the organic buildup that caused the hydrogen sulfide buildup. And then, so they switched to a floating feed, and this seemed to alleviate the problem. There's still some problem, but not as much as they were seeing before during harvest. And so in bait fish, we have noticed that large amount of fish around the springtime will disappear from the pond completely. And what seems to be happening is hydrogen sulfide is building up, it's killing the fish, and before they don't float to the surface, they get decomposed, which just causes more problems, and so it's disappearing fish. And so some of the recent research was conducted to rehab your pond, which is one of the topics, is to rehab. One of the ways to do it is to add aeration to your pond. If you add aeration, you increase the dissolved oxygen levels, and this decreases the toxicity of hydrogen sulfide. Another method was ponds that weren't drained or renovated every 2 years or so. The buildup would be so great that the hydrogen sulfide was causing problems in their ponds. So they found that if you drained your pond, you till up the surface, and this exposed the hydrogen sulfide in the water to air and oxidized it to non-toxic forms. And in the past couple years, Dr. Kelly was looking at hydrogen sulfide in aquaria and using pond soils, building up the hydrogen sulfide to low, medium, and high toxicities, and adding what's called Paracoccus panatrophus, which is a bacteria that is in the product, the pond detox. And when they add this to the pond at the level that's recommended, it's recommended at 400 grams per acre. And when they added it, within 24 hours, the hydrogen sulfide levels were significantly diminished or completely taken out in the low concentrations. And so that was the preliminary studies. That was in tanks, so they changed to see what the difference was when you come from a tank to an actual pond. And that's one of the things I'm looking at now is we're adding hydrogen sulfide in bait fish and sport fish ponds, or not hydrogen sulfide. We're looking at hydrogen sulfide in bait and sport fish ponds, adding pond detox at a control of 0, 100, 200, and 400 grams per acre. And they add it in the spring when the water temperature gets to about 70 to 75 degrees. And you add it, once you start adding it, you add it every 10 days or so for about 4 months until the waters get below the 70-degree mark again, because that's when problems don't seem to occur as much. And so I'm currently looking at the effect that this pond detox is having on hydrogen sulfide, and also looking at one of the side effects it seems to be is blue-green algae. And blue-green algae is very unsatisfactory for aquaculture ponds because they do not produce oxygen quite enough, and they also have boom and bust cycles. So they'll get a lot of algae in the pond, and then all of a sudden they crash, and the dissolved oxygen levels will crash as well, especially in the morning hours. And so that's why they decided to use the aeration. They thought it might be a method, but it seems to be costly because you have to have your aeration on constantly. It costs a lot of money to run those motors. And so we're also looking at blue-green algae in accordance to the pond detox. And also, hopefully, they seem that the product claims that in shrimp, when it was developed for, that yields and survival and growth was much higher. So we're also looking at yield and survival in these ponds, trying to see if this could be a bacterial amendment that can be used in aquaculture.
Underneath this sort of more secure area, this fenced area, we've got a lot of bait fish studies that have historically been going on over the course of time here. Bait fish, as you probably know, they're pretty susceptible to birds, and on most days you'd see bird netting over the top. About a few weeks back we had a major blow come through that took the bird netting off the top, so we're working to get that taken care of. But up here we're going to start seeing ponds that look a little bit different. Actually, you can see a pond over there that looks a little bit different with some paddle wheels in it. This is sort of a alternative means of raising fish called split pond design. It's been made more famous in catfish ponds, and we'll see a demo of that on the other side. Dr. Nathan Stone has started to look at using split ponds as a means of raising bait fish. And the next demo is going to be Dr. Stone talking about some of those results. But if you look at these ponds, what you end up seeing is on the other end of this pond, you've got the fish rearing area, and then the water ends up being pulled out, and this is the water treatment area. So you have, the theory is that you have better control over your fish, and probably the theory also is that you can end up feeding them a little bit easier. The jury's still out, and Nathan has been doing this pretty much non-stop for the last year, trying to get this some data out for you guys, trying to get this up and running. Dr. Stone, many of you know, has been here for years, and this is just sort of the latest idea with respect to trying to adapt the industry to the future, making it more productive.
What you're looking at is a 12-pond study comparing the growth of golden shiners in regular ponds, earthen ponds, and split ponds. This is the third one we've done in here, and this is during the growing season. This was stocked in early July. And Kayla McCoy, Matt Smith, and Sindhu Kamal have been taking care of this study, and we'll pull it out next week if everything goes well. This is basically, the paddle wheels kick on at 8 o'clock in the morning. They go off at 10 o'clock at night. The fish are all on the inside of that fence. Ordinarily in split ponds, it'd be commercial ponds, it'd be a solid earthen levee. In this case, the fish are all confined on the inside. Then at night, at 10 o'clock at night, the paddle wheels kick off, the aerators kick on, and the fish have good oxygen for the night. And then in the morning, the cycle starts over again. This was basically the same design as developed by Dr. Craig Tucker at Mississippi State. We're just trying it. This is sort of the first trial on it. Share some of the same advantages. It's easier to feed them. It's easier to aerate, if you think of large ponds anyway. It sure is easier to harvest. We definitely noticed that in sampling. The disadvantages are also there. When you're dealing with minnows, they're really small. And so the screens that you have to have are correspondingly small, smaller, fewer open areas in it. You have to push more water through. This really won't work in the spring when they're spawning. You know, the minnow fry, they just spawn and spawn and spawn, and there's lots of little babies, and they go right through this fence. So unless you have a predator or something on the other side that clean them out, it's going to become a regular pond in no time at all. The biggest advantage may be for fathead minnows, that really it's more of a feedlot kind of a fish, and they don't feed well in large ponds for some reason. That I think they're kind of stupid. You have to get the feed on their heads, and even then they don't really do it. One of the differences with catfishing, you know, in catfish they feed a lot, and so this whole area here is called the waste treatment area. It's for processing nitrogenous waste. Well, in baitfish we don't have that. The feeding rates are really quite low. So it's really natural foods production. And in minnows, at least half their growth comes from natural foods. And so what we were hoping was by excluding the fish from this part, we'd actually have like a zooplankton refuge, and we push that in there. But if you don't have enough flow, you're going to reduce your growth. And that's what we've seen in our sampling, is at least in the lower density, they're doing a lot better in the open pond. There's not as much of a difference at the higher density. So we're going to have to do some fine tuning, push more water flow through or something like that. Anyway, that's about it.
Well, the split pond idea has taken off on, as far as a lot of the research that we do. A few years back, Craig Tucker started down at the USDA shop, started doing a lot of research on this. We started working in parallel, coming up with alternatives to that. Ultimately, I think we've come up with a design, when David Heikus was here, that takes into account some of the things that the farmers want. The next design is essentially a split pond design that is set up for catfish. And what we're doing is retrofitting a lot of these ponds so we can have some on-station experimentation with split pond systems and comparing it to the typical pond construction. As you probably know, we've been doing a lot of economic analysis over the years, and certainly with what Dr. Engel has been spending a lot of time is different variables, different sizes of classes of fish in terms of stocking densities, and incorporating all that data into economic models. So ultimately having this facility here is going to end up sort of becoming a next generation test facility with respect to comparing split pond systems to conventional pond systems. Where it works, how it works, how it can work better. These are all questions that the industry is asking us to participate with, and it's a very important component with respect to the type of work we do down here on the station. This next display is Dr. Jay Park. He is essentially an aquaculture engineer, which, as you can sort of appreciate when you start putting together some of these systems and designing pumps and flows and infrastructure to move water that lasts in a production environment. Having an engineer like Dr. Park is very important.
Good morning, my name is Jung-Hwan Park. I'm an aquaculture engineer. The topic I would like to talk about today is the fundamentals of the split pond system. As many of you already know, partition to aquaculture systems and its variations are getting popular in the catfish industry in the United States, and it suggests a new paradigm of pond aquaculture in the United States. So usually what you have today here is one of the variation of those systems we call split pond systems. So usually the split pond system consists of two uneven sections, which is divided by the earthen levee, and usually combines the fish in smaller area. And of course, this is not the right split right now. This is engineering pond, so it has a 50-50. But usually, it has a 20 versus 80%. So 20 is the fish basin. So usually we confine fish in the smaller area that comprises about, like I said, 15 to 20% of total pond area. The remaining area is 75 to 80%, and that is the kind of water treatment, the section, which provide the oxygen and treat all the poops and pees of the fish and like that. So what we have here is, during the daytime, that circulator pushes the water into the the waste treatment, and then that water, sitting in that, basin, going back to the fish basin. Why it happens, the ammonia nitrogen, which is the waste material from the, fish, will be removed in this section, and then relatively cleaner water will go back to the fish basin. By doing that, actually, we can crowd the fish in the small section, and it gives us a lot of the, beneficial things, like imagine that we have a really huge area, and then we have a less number of fish in there. But rather than that, we crowd them into smaller section, and it gives us really a lot of the convenient stuff, like easily we can feed them into the smaller section, and then we can easily aerate in the small section. So during the night, we can keep the fish survive very easily. But sometimes the bad thing can happen because we crowd the fish in the small area. So that means that they, consume the oxygen very quickly. So without the, proper aerations during the night, we could have a really high likelihood to kill the fish at one time. So that is kind of a drawback of the split pond system. But in general, we got a lot of beneficial stuff out of this kind of system. So for 2 years, we gathered some informations on commercial split pond system to figure out what is the proper engineering criteria we should go with. So the extension agents and research personnel gathered a lot of the information about water flow and the power consumption according to the RPM, right? And also the water depth. Those are kind of key parameters to determine the performance of the paddle wheel. Why that is important? Because as you can see, like I said, this is a not right ratio, but the main investment portion is from building of the levee, and then fabrication of the that circulator and other related structures. So easily it can go up to $10,000 per 1 pond, 5 acres or something like that. So those two things are the main portion to eat up your money if you want to go with the, So that's why we try to come up with some kind of the numbers out of that, paddle wheel to generate the engineering criteria. So what we found out was, of course, when we turn it around faster, we're going to have more water pushes into the waste treatment. That means we could have better efficiency to remove the fish waste. But in that case, we're gonna eat up a lot of the power consumption, which is not really desirable. So another thing we have to consider with the higher RPM, it causes a lot of the friction, right? And then it gives a lot of torsion and tension on that shaft. So sometimes it can break, right? So what we're trying to do is reducing the RPM. But for that, to have equal amount of water with the reduced RPM, we have to do something on it. So that was what we're trying to find out. So what we found out was, increasing the RPM, causes a lot of the torsion and tension. Sometimes breaks the shaft and eat up a lot of the the power consumption. And increasing RPM, the power consumption actually increased exponentially. But at that time, the water flow is not increasing that much. So rather than that, we better increase up the width or the surface area of paddle wheel to contact the water, right? And then, but at the given water depth, it is not practical to have a infinite width of the paddle wheel. So that's why we try to come up with some kind of numbers out of that. So what we found out was, obviously, increasing increasing the surface area of the blade is more efficient than increasing the RPM. But at the given water depth, it is not practical to increase the width of that. So in general, in total, we should go with like a 4 RPM in rotational speed. And then based on that thing, after 4 RPM, actually power consumption dramatically increased up. So it's a kind of a 4 RPM is a threshold we should keep. So below that one. But if we go below that one, that means we will have the wider width of the paddle wheel. So I think we think the 4 RPM is kind of a threshold. And based on that things, right, we should determine the the total surface area of the paddle wheel. So that is kind of a things we come up with so far.
Yeah, we have some broodstock studies going on. These smaller ponds are getting set up right now to stock probably next week for a feeding study that we're going to be doing with broodstock. Feed them through the winter with different diets, and then next spring we'll end up spawning them and looking at fecundity and other indices of fry quality. So we've got 9 ponds here that will end up being stocked, 3 ponds per treatment, and ultimately this experiment is one of the longer ones. It won't get finished probably until July of next year. All right, coming up next is Dr. Anita Kelly, and she will be talking about crappies and specifically different ways of producing sterile crappie. A lot of you may know that crappie have a tendency to overpopulate things very quickly. One of the ideas is to try to produce sterile fish, and that way they would end up not doing that. And I'm going to, essentially let Dr. Kelly sort of describe what's going on.
Anybody on here a producer? Okay. Anybody interested in raising crappie? Everybody like crappie? You like crappie? Okay. Well, if you know crappie, you know they're hard to raise. And so one of the things that we're looking at is using them as an alternative species for aquaculture. Well, in order to be able to do that, we have to overcome one important thing, and that's the amount of reproduction that they have in a pond, because they have a tendency to really be proliferative during certain years. So in order to do that, we can do a couple different things. The first thing we've done is actually made a hybrid. Now, when you make a hybrid between a black crappie and a white crappie, it grows faster than either of the parental species. We can also make them triploid, which means that they'll be functionally sterile. But the induction of triploid in a hybrid actually makes them survive. The survival rate will go up on them. For some strange reason, nobody knows why, but that seems to happen. So we've been working on this, and we actually have had some pretty good luck with getting them raised up, getting them feed trained, and stocking them out. And one of the things we thought about is there's been some research that was done a long time ago where if you look at F2 hybrids, if you take the original hybrid crosses and cross them again, you get what's known as the F2 hybrid. And by doing that, you get really, really low reproduction in a pond, so you wouldn't have to do the triploid induction in them. But the problem with that is that you lose that growth that you get with hybrid vigor. So we've been actually looking at just doing the F2s this year, and we've had pretty good success with the F2s. The nice thing about those is we can put them in a pond and let them spawn, and we don't have to take them out and worry about them and have to manually strip the eggs and the sperm from them. So that's kind of where we are with our crappie. We've been able to get about 4,000 pounds to the acre so far. We haven't pushed it any higher than that, because if you've ever dealt with crappie at all, you know that when you handle crappie, they just seem to kind of want to die. So they're kind of a cool crop that you'd want to only handle when it's cool out. But we're going to go to work on getting them at higher densities in the ponds. Now we have feed trained them by putting them into little blue tanks along the sides of the pond itself and actually concentrating zooplankton and feeding them high concentrations of zooplankton before we put them in the pond. And that's also increased our survival rates in ponds, because sometimes you'll stock out larval fish, and in one pond you might get a really nice crop, and in the next pond you might get nothing. But we haven't had any complete blanks yet doing the zooplankton treatment beforehand.
Well, that's the last formal demonstration. There are a variety of other things. The walking tour is something that I would encourage you all to take part in. But just as we go back, I mean, you can certainly tell that there's been a lot of construction, particularly in this section of the station recently, in terms of changing what we had into a development area for this split pond technology. And a lot of earth-moving equipment running around. Over there, a variety of equipment that we are still using in terms of specific experiments. A lot of you remember David Heikus in terms of building various things that the industry needed, and whether it's net reels or bar graders for hybrids. A lot of that stuff came out of the shop area up here, which Dr. Park has kind of inherited. We've got a new hydroponics facility that's, don't have a display there this year, but hydroponics using the effluent water to raise vegetables, flowers, that kind of thing. A lot of people in the area are becoming more excited about that, particularly schools in the area are becoming excited because it's a good science experiment. And again, I certainly appreciate you coming in, and we like showing off. We feel proud of what we do, and we are in fact trying to stay connected as best we can, and hopefully this kind of prompts some ideas where you can say, hey, what if we did this, or maybe we should alter that. Well, that's sort of, we've got a lot of folks vested in our extension program in doing just that. So I appreciate your attention, and hopefully have a good rest of the day.
So what we have here is a bunch of largemouth bass in tanks, and we did an experiment last winter looking at growing fish indoors in tanks. A lot of largemouth bass have been growing for sport fish a lot of time, for stock enhancement, so growing up small fish and then turning them into lakes and such. But there's becoming more of an interest in producing largemouth bass for food fish. And so Arkansas currently is the largest producer of bass for food fish. And states like New York have just loosened up some regulation on selling bass as food fish. So what we wanted to look at was raising fish indoors. Typically, bass is about a 2-year product. So in the first year, we're spawning the fish, putting them into ponds, growing them up, and then in the second year, finishing those bass out into a food fish product. And so what we wanted to look at is if we brought these fish indoors in the fall, spawn them in the fall, bring them indoors, try to get a head start on the first growing season and see if we could shorten that overall time. And so what we have in these tanks, you can see, is we looked at different stocking densities of these fish, and so you can visually, if you look, see how crowded they are on one end and how low they were on the other. It's kind of generally been thought that because bass is a predatory fish that maybe they wouldn't do so well in tanks, but really we found pretty good overall performance in all of these trials. So we started with a 3.5-inch fish, grew them for 60 and stocked them at different densities, grew them for 60 days, and evaluated them. And so we grew them from about 3.5 to 5.5 inches in that 60 days. Then we took them out, resorted them, put them back in at 5.7 inches and grew them out for another 60 days, and they got to about 7 inches in that period. And we really had, in both the first phase and the second phase, saw pretty similar results as far as feed conversion, growth rates. They performed well in all the treatments. We had high survival rates in all of them, as high as about 97% survival. And so really it was a success. As we started getting further along, we did see that in some of the higher stocking densities, the fish developed some lesions on their skin, and kind of the sweet spot was this fourth tank here. While we raised at the maximum fish to a little bit above 1 pound fish per gallon, we found that about 0.7 pounds per gallon is where we saw the best results in the system. And so this is a recirculating aquaculture system. I don't know if you're familiar with that, but basically, so we raise the fish in the tanks here, and then the water moves from these tanks into this settling chamber, and so this allows any uneaten feed that's been pulled off through the side drains or the bottom drain, feces, any solid waste to kind of settle out, to try to get rid of some of that before it comes into this next chamber, which has this plastic media in it, which allows bacteria to grow on it. And with these bacteria, what we're trying to do here is get these bacteria to take up the ammonia from the water. And then from there it moves through into a pump through a sand filter, takes out a little bit finer particles from the water, and then goes into this tower here where we inject pure oxygen. into, so that's just an oxygen contactor. And then from there it goes through some UV bulbs, which helps to sterilize the water as it moves back into the tanks, and it just circulates round and round and round. Yeah, just keeps going. And then we've got some heaters over here. We kept the fish at about 97 degrees the entire time. And then up on the wall up there, we've got that box with that blue cord, has an oxygen sensor on it, which is hooked up to an alarm system. And so we set that to say if the oxygen drops below a certain point, go ahead and give me a phone call, even if it's 3 o'clock in the morning, so I know that we can get in here and get something taken care of before we find dead fish in the morning, to keep tabs on everything. So really, it showed a little bit of promise that, bass can be growing. I mean, there's still more research to be done as far as different temperatures and looking at different factors, but overall, we were quite happy with the results of the study.
So the goal was to get them to a certain size and then put them in ponds outdoors to finish them.
Yeah, exactly, and to try to shorten that process, because now it's about a 2-year process. So to get them to the stocker phase that you would go to grow out we spawn them and grow them up in the first year, then they're overwintered in the pond, and then the second year is for that growth phase from about here onward. So if we could condense that first bit in tanks and see some good growth, then we could hopefully shorten that growing cycle to maybe into a year, or maybe a year and a half, depending on
quicker return on your investment.
Exactly, and so the idea is to get those fish in, grow them up, and get them off to market a little bit faster.
Now, is there anybody that's looking at, instead of this fancy recirculating system you've got here, combining the hydroponics with the recirculating system?
Yeah, so hydroponics is kind of a breed of, and we've got a greenhouse out here where we've got some aquaponics or hydroponics going on, going on. I'm not sure if Rex has raised some. He did have some bass out there, so he's worked with some different species, and it very well could be, because we can raise them in tanks here. There's no reason that we couldn't raise them in tanks and use their waste to grow plants off of them. But we do have some of that research going on as well, and folks looking at sort of the aquaponics, and there's been a lot of interest in that as well.
This stop here is to talk about a project that we started earlier this year. About 2 years ago, fish farmers started reporting a lot of losses, which they thought were from birds. The number of acres in production in catfish in this state has been reduced and converted to row crop. So those ponds aren't around. So what's happening is you're having what we think is a concentration of birds that are on fewer ponds. The same number of birds are still coming down here. There's just less water for them. So farmers had been reporting that these lesser scaup, which is a diving duck, had been eating quite a bit of fish. And a lot of these guys will lease their properties to duck hunters during duck season. And duck hunters were shooting these scaup and finding fish in them. So they wanted to add them to the depredation permit, but the Fish and Wildlife Service didn't want to add them without evidence, that they were eating fish. So that's why we basically did this project. We got a permit from the Fish and Wildlife Service and from Ag FC to look at. We got a permit for 200 ducks, and we killed 120 of them. What we would do is we would wait until they were foraging on the ponds of what we believe fish, and for 10 or 15 minutes before we shoot them. The reason for this is that ducks can digest the contents of their guts really quickly. So within 30 minutes, according to one duck biologist I talked to, you won't find hardly any trace of fish in the duck. So we collected them, came back to the lab, and as you can see here, this is a scaup that was dissected. This is kind of the top of the esophagus coming down from the throat. And then this here, where all the fish were found, is the crop. And then you have the gizzard down here. This is the contents of the gizzard, which the duck, it's just like a chicken gizzard, it uses to grind up stuff, and then goes into the intestine. So, we did find in almost 11% of the ducks that we collected whole fish, and sometimes we find 18 to 20 fish in a single duck, which is quite a bit of fish. And we're also looking for fish bones in the contents of the gizzard and also in the intestine to try to get an idea of what percentage of the ducks are consuming fish. And what we really want to know is what is the weight of the fish inside the ducks, so we can kind of get an estimate of how many pounds a farm might be losing in a given season. And what happens after duck season is these ducks are going to migrate back up north, and so they need to really eat a lot of calories. And that's when we're seeing more scaup on these ponds becoming more of a problem in February, March, before they make that migration back north.
Let's first talk about intensive culture systems. We had to change over to these systems, or we're making some changes in the industry right now. We feel like we've got to increase the profitability for the farmers, and that's lower that cost to raise that fish. One such system is called a split pond. Another system we have is called an intensive aeration system, and we also have called an in-pond raceway system, which currently we have the split ponds in southeast and northeast Arkansas right now. Several of those units up and running. In fact, they've been running for the last 3 or 4 years. We have an intensive aeration system in Ashley County, just over the Chico County border. 6 units there, and we no longer have the in-pond raceway system, which we once had. But if you come look at this, you can see why it's got the name split pond. We started out basically with a 10-acre production unit, and in this situation, you want to grow the fish in a concentrated area or about 2 acres of that pond. Well, what might be the advantages of using this system, you think?
It's easier to harvest.
Well, it's much easier to harvest, doesn't take near as, equipment. In fact, when I was young, years ago, as you can tell by my age, we had 40-acre ponds, and these things were square, and you would have to have nearly a mile of seine on that seine reel the time you went around the corners. 1,320 feet across 40 acres, but then you'd want a little bit of bow with the seine. So you're talking about tying nearly a mile, or have a mile of seine. You might not have a mile of seine reeled out. But anyway, we feel like we're concentrated the area where we feed. In the typical day, this thing is running, this little thing is running. Oxygen is produced here. Your waste that your fish give off in the form of ammonia is cleaned up, and you're introducing clean water back in here. We found that these units are very productive, 15,000 pounds per acre here. And I'm talking about as a whole. We're talking about 150,000 pounds per acre versus a traditional unit of about 5,000 pounds per acre production. So they can be good, but again, you've got to go strictly with hybrid catfish. The regular channel catfish can't handle it. But with any system, you intensify it, you're going to have problems, and these fish are going to get sick occasionally. I'll give you an example. When I was younger, managed farms, we had 20-acre ponds. We wanted to treat a pond. We had to get the chemical, put it in our boat in what's called a chemical well, and we would drive the boat through the pond, do a transect down, over, a transect up, down, across, and then go crosshatch. We didn't get good dispersion because we would have very hot spots, and we'd have cold spots, and hot spots and cold spots. We feel like this type unit that we can shut this aerator off right here, go ahead, put our chemical out, the same means as we put it out years ago with the chemical well. Then we want to turn this on. Turn it on, let the chemical mix. Start this at 8 o'clock in the morning, let's say. Let this run for the most part to about 4, maybe 5 in the afternoon. Then turn this unit on. Let the water flush through overnight. You feel like you've got your treatment concentrated. Now, one advantage, as she mentioned, it's cheaper. The research that Ganesh Kumar did here at UAPB, using a 4-part per million potassium permanganate treatment, there was a saving of 79% in this unit, or the split pond, versus the traditional unit. The chemical cost was told about $2,100 for the chemical. He did it for like $400. That's 79%. So that's very good. And if you use even more expensive chemical, the savings are even greater. Other units that we talk about are intensive aeration, where think of this as a 4-acre pond. We would have three aerators, about 35 horsepower aeration. We found out that we can grow 12 to 14,000 pounds per acre in those units. Again, they're concentrated. Again, growing the hybrid catfish. And in-pond raceways are a little different. All right. Think of these as being about 1/4 of the size, having 4 cages that are about 80 feet long by 20 feet wide, stocked with fish. Can be tilapia or any kind of fish, but occasionally they get sick. So what the producers do in this area, in Alabama where they run them is have a weighted tarp, and that's talking about a weighted bar on the tarp. Just reel the tarp down almost like an awning to keep the water flow from going through the cage. Put the chemical out, let that run in there for a couple hours, turn your aerator back on and flush it out, and get a concentrated equipment with concentrated treatment, and get the chemical out in a hurry, so you get much more concentration and then probably better effect of treatments. These things, again, we've got 4 of these. I'm sorry, we got more than that. We've got units in southeast Arkansas and around Newport in northeast Arkansas. Maybe more of those to come. Other thing I want to talk about is Eromonas. We have a strain that showed up in Arkansas a few years ago. Eromonas is a catfish disease. We call it the hot strain because it is more virulent than our typical strains of Eromonas. It can get spread, and be devastating to catfish. So we've got a treatment protocol we suggest. If you're buying fish from any farm, send that sample through our disease lab. We will do culturing, determine if that's present. If you're buying those fish and we've determined that they're good, go ahead and stock them in a pond. Isolate that pond for about 2 weeks, which means don't share equipment between that pond and other ponds. Watch those fish. If you're buying from a farmer or a finger producer, make certain that they wash their hauling tanks out very well before they put your fish on. We actually got this disease, or it could have been spread from Alabama by hauling from an infected farm. That's one of our theories anyway. But we feel like if that hauling tank is washed off the truck, any kind of equipment such as seines, dip nets, boats, baskets, anything used in harvesting, make certain that's disinfectant. We have a disinfectant we use around UAPB, our station here, called Virkon. Just mix a little in like a 2-gallon pump sprayer. Spray the equipment down, the waders and such. Let it dry, and that will clean that off and make that safe. As far as a seine, if you have infected ponds on your farm, you probably need to have one seine dedicated for the ponds where the disease is and another seine for the other areas. But after you use a seine, be sure to stretch that seine out, let it dry thoroughly. And I'm not talking about 2 hours, I'm talking about 2 days, 3 days to completely dry through and through, because if you look at the bottom of the line of the seine, you've got a maybe 9 or 10 up to 20 small ropes with lead weights on those. So they're quite dense, and mud can get in there, and sometimes they don't dry well. So you want to get it as dry as possible. As far as aeration equipment, you don't want to take aeration equipment out of an infected pond and set it in an uninfected pond. If you do, get it out, get your Virkon, spray it off. It may take a little while, but it's better to lose a few hundred fish than a few thousand pounds of fish by spreading a disease. If you do contact this disease or think you have a disease on your farm, get one of our diagnostic people. They're listed on here. We have the UAPB lab, Lake Village, Lone Oak lab, and our person at Jonesboro. We get out on the farm for you, and we will actually do the culturing on site. And we want to get on this situation or get your help you with your situation as soon as we can, so your fish will start healing.
Welcome to the Fish Nutrition Research Program. This morning we're focusing on trying to make diets with more plant proteins in them for bass. And bass, of course, are carnivorous fish. They don't naturally eat much plant material. So the challenges are twofold. One is to put plant products in the diet that are nutritionally appropriate for bass. And the second challenge, which sometimes turns into the first challenge, is getting them to eat it. So we can make a diet that we know meets all their nutrient requirements, but if they won't eat it, it's not helpful. And the reason we're trying to increase the plant protein content is the traditional protein source for bass has been fish meal, marine fish meal. And it's an excellent protein source. It's like our gold standard. It supplies everything, and it's very palatable. But it's based on natural fisheries, and levels of natural populations have kind of leveled off. And it has become extremely expensive to use marine fish meal and fish oil in the diets. So cost is one of the primary motivating things, but the plant products too are seen as more environmentally sustainable to use. So if we can increase those and minimize the fish products, then it should be better for the farmer. It should be a more profitable production as long as it doesn't reduce their production. We have a couple of studies here. We're collaborating with a number of different people. The study on the right is a hybrid striped bass study, and we're doing that in collaboration with Stuttgart. And also, a scientist at Fayetteville has produced some new strains of soybeans, and this is through traditional breeding. They're not genetically modified, but they have lower levels of anti-nutritional factors, which is one of the main limitations on using plant products, and they also have quite a bit higher protein content. So the first step that we did with those new strains of soybeans, we had to get them processed so that they were commercially similar in form to the stuff that we normally use. Then we did digestibility trials with the fish. So we measured the nutrient digestibility of these different soy varieties, and we compare that to digestibility of fish meal. And also we have like a commercial soy protein concentrate in there, because these products have been tested and we have a track record with those. We're finding that in general the protein digestibility of all the soy products is good relative to fish meal, but the dry matter digestibility, which is kind of an overall digestibility measure, is lower in the soy products than it is in the fish products. So we may have to do some further processing to bring up the quality of the soy meals. Then the next step with those is to formulate diets based on the nutrient digestibility and feed them to fish in an actual feeding trial. If there are problems there, then we may have to add palatability enhancers to the diet. We can use different kinds of amino acids and things to make the diets taste better for the bass. So assuming we can get them to eat them and they work out nutritionally, that should reduce feed costs and it should increase the profitability of the farm. With the largemouth bass, we're doing something similar. In this case, we're collaborating with a food scientist in Fayetteville who's also altering soybean meals through chemical processes, acid hydrolysis, and just warm water. And he's been able to increase the protein content by about 20% on different soybean meals, and also reduce anti-nutritional factors at the same time. And in this study, we're also testing some fermented soybean meal. Fermentation, if you add bacteria or fungi to the soybeans, it starts to break them down, but sort of in a controlled way, and it actually ends up increasing the protein and decreasing some of these things the fish can't digest. So we're just at the stage right now where we just developed these new soy products, and we're making the diets now to start a feeding trial. But the goal for both of these fish is not necessarily an all-plant diet. We're perfectly open to using other animal products like the poultry meals. There's a lot of poultry meal out there, and it's tried and proven. It's a good protein source for fish. So plants in some combination of animal products other than fish is the most likely route to go, and to make the diets more environmentally, I guess, acceptable to use long term.
Remember, if we go, for example, in housing market, either we are buying house or selling house, our realtors say, oh, probably the price of your house is $200,000. They essentially estimate the price based on some kind of a mathematical model. That model determines price of our house depending on how many bedroom we have, location of house, school districts, this and that, whether it is brick or is it some other kind of material. Same is true for fish or aquaculture products. So whenever we are buying catfish or salmon, we pay not for only salmon, we pay for even the level. For example, I normally pay higher price for a commodity that has the expiry date, because I do not like to pay for a rotten product. Right? So I'm very particular to see whether that product has a date of expiry. So those small things leveling also do matters, right? So what we have here is two cases. One is the bait fish case, another one is the catfish case. The bait fish work is mostly done by my colleague, Mr. Morales, and the catfish work is mostly done my colleague, Ms. Oli Chen. So we developed two models. For bait fish model is based on 203 bait fish retailers. We conducted survey across the United States and find out what kind of products are being sold and what kind of price consumers pay, wholesale level as well as retail level. For catfish, as I mentioned earlier, it is based on scanner data. Now, what is the scanner data? Scanner data is, for example, take the case of this water bottle. Whenever I go to Walmart, what I do, I get this and take it to the counter lady or gentleman scan this barcode, and it shows on a particular day a particular bottle of this great value, how many size? 1 pound water bottle was sold at the price of this. Then some companies buy this data. And we as a researcher, again, buy the data from them, because that shows, okay, what determines the price of this water bottle. So for fish, we could find out what determines the price. And that's why I think you came, and then we talk about this result. So what we find here for catfish model is that though there are a lot of products in the market, but there is a concentration. People, not all products survive. Think about it. That's essentially the case. We might be producing a lot of product, but few products are liked by consumer. As a result, it survives in the market. What we are finding as a result of that is that in spite of a larger, relatively large market volume, it is concentrated in few species, few brands, and few products form. Meaning those are well liked by consumer. So if we are developing a marketing strategy, we need to know what are those things. And you'll see here, we find that species do matter. We found some commodities like tilapia, swai, flounder, whiting, they are capturing about 65% of the market share. And as I mentioned earlier, packet size matters. Smaller the package, usually higher is the price. You command a premium price for a smaller market size. Some of my industry people might say, oh, this is more expensive because I now need to have individually packed. But think about it. If it costs you 1 cent extra to have a smaller packet, but if you can get 2 cents more by selling, why will not you do that? So we need to see what consumer pay. And our data shows, our model shows that, yes, smaller the size, it commands more price, premium price. But it varies across the region. There are regional specificity. And the kind of value addition you do, whether it is smoke, whether it is season, also matters. And earlier, my colleague, Ms. Chen, was saying that, think of the case of the basa and swai. In general, it commands lower price compared to U.S. farm-raised catfish. But when they smoke that product, you command higher price because of the smoke attribute. Salmon is a case, they command higher price. This is true also, for example, for many other commodities. And here what we find is that the private brand, like Walmart, if you are marketing your product through those private brands, it commands higher price.